This video is cytoskeleton microtubules GTP cap. Here's some key background information to jump into this topic. First off, tubulin dimers are enzymes. We've talked about them as globular proteins, but they have enzymatic ability. And in particular, we're going to focus on beta-tubulin. Beta-tubulin can bind to and hydrolyze GTP into GDP, and each tubulin dimer binds to one GTP. Again, we're going to focus on beta-tubulin here. But you may run across it that alpha-tubulin also binds, but we're really going to focus on the beta-tubulin part. So when beta-tubulin hydrolyzes GTP to GDP, this is typically shortly after it's been included into the growing microtubule. And then GDP remains tightly bound to beta-tubulin. A couple things to note here. So the GTP cap is not an extra protein. It is instead just the tip of the rapidly growing microtubule, which is made of GTP tubulin dimers. And then also watch out for this. This GTP cap is not a microtubule capping protein, although it sounds like it is. Bad naming, but that's the deal. And this enzymatic property is what underlies the dynamic instability of microtubules. This is figure 17-15 from your textbook. So let's break down this figure. Basically, let's look at the color coding. So over here, you see these free-floating or cytosolic heterodimers. And notice that there's this light green, which I've actually put in yellow, and that's the alpha-tubulin. And then we've got this kind of olive-looking thing with a green rim and then a red center, and that is beta-tubulin bound to GTP. And then as we see a lot of those here on the growing microtubule tip, this is what the GTP cap is when we have a high proportion of GTP-bound beta-tubulin. And then as we look further down the microtubule, this is where GTP has been hydrolyzed by beta-tubulin, and now instead of seeing these red circles, we just see the dark green solid circle, and that is GDP bound to beta-tubulin. So fun fact is that about half of the tubulin that is found in the cell is in this cytosolic free-floating state, and about half of it is found at any one time incorporated in a microtubule. As we'll see, the free-floating heterodimer is going to have GDP, but it will pick up GTP quite rapidly when it's free-floating in the cytosol. Okay, so I have a few notes to keep track of on the growing microtubule. And so basically here they are. So the tubulin dimers are added faster than GTP can be hydrolyzed to a growing microtubule, which facilitates the GTP cap formation. And again, this is always going to be on the plus end of the microtubule. Also note that as far as thermodynamics, that the GTP-associated tubulin dimers are bound more strongly to each other than the GDP-associated tubulin dimers are bound to each other. And so basically what that means is that this end is more stable than this end. Okay, now let's talk about the shrinking microtubule situation. So if we go up here towards the top to check out this figure, then notice that there's no GTP that is bound to the tubulin. So this is all GDP bound. And the protofilaments, remember those long strands, those are going to peel away from the microtubule wall. And this is really cool videos to watch, like on the inner life of the cell. So this just kind of peels back. And basically the GDP tubulin is released to the cytosol, and so as I was mentioning before, that's the case where this is destabilized, and then GDP can pick up GTP pretty readily and then go back over and be incorporated into another growing strand. Okay, so let's check out the notes here. So basically, for a shrinking microtubule, on the plus end, there are all GDP bound dimers. These GDP bound dimers do not tightly bind, and so disassembly is favored at that end. And the cytosolic GDP, which is bound to tubulin, will rapidly incorporate a new GTP and then can go join another growing microtubule. And basically you can see how the state of either shrinking or growing is favored by the presence of the GTP bound tubulin or the GDP bound tubulin. So this can really escalate the growth, and this can escalate the shrinking. This figure is not from your textbook, but I thought it might be useful to see a different way of looking at the same concept. Also, this incorporates two other terms, which you'll see occasionally show up with microtubules, and that is catastrophe and rescue. Kind of dramatic terminology here. Basically, what we're seeing here in gray, this is a microtubule, and then they are symbolizing the GTP cap in dark green. Remember that these are still just the regular old heterodimers, but in this case, beta tubulin is bound to GTP. And they're showing that overall that the microtubule would favor growth in this case. And so we see those little microtubule heterodimers being added here in these little circles. And that's not exactly how that happens, but the concept is that it's growing when it has a GTP cap. Now, if there's an accidental loss of the GTP cap, due to kind of chance by dynamics within the cell, they call that a catastrophe. Again, dramatic. So now the GTP cap is gone, and basically this leads to rapid shrinkage. Then, at some point, as we know from dynamic instability, then this microtubule could start to grow again. And the reason that it would start to grow again would be because it would regain its GTP cap, which we see here. And then as we look down the figure, then we see that more heterodimers are added, and so then we can see that it continues to grow on that plus end. And so this is called a rescue, when it regains the GTP cap. Basically, catastrophe and rescue are just some kind of fun words, if you will, to describe this dynamic process. I want to put in a plug for the movie 17.3 from your textbook. Also, this really showcases how GFP tagged proteins can be used in two different ways to examine dynamic processes. And this plays off one of my take-home messages, which is that cells look cool, and in particular, this is due to fluorescence. So we've got green fluorescent protein, that's GFP, and so basically that's been tagged in one experiment here on the left to a protein called EB1. And so if you reach back to the FRAP video lecture, we talked about GFP tags and how this is a naturally fluorescing molecule. This in the video you can see is a very dynamic process, and so it basically looks like some sort of meteor shower is taking place within the cell. And so this is one sort of label for microtubule processes. And then over here, we see a different GFP tag protein. This is GFP tagged to tubulin. And so you can see that the pattern of the fluorescence, which is shown here in white, is very different. Not as many like kind of like shooting star looking things, but instead like big long streamers all over the place. And so from what we've talked about so far, can you think about what might be labeled here as far as what part of the microtubule is labeled versus this part here? And so the answer is that EB1, this is part of the GTP cap. And so basically we can see just the GTP cap for growing microtubules in this part of the video. And whereas over here we're labeling the entire microtubule, whether it's growing or shrinking.